lecture we have discussed about the microsomal enzymes, non-specific type of enzymes that generally acts on diverse functional group of drugs, diverse category of drugs. That means these are non-specific in the sense that they can act on different classes of drugs, non-specifically. There is no specific point that the drug will act on some certain special kind of mechanism, or it will generally adopt something very of specificity. Okay, so lack of specificity, but still we can categorize the phase 1 reactions into different types, just like oxidation, reductions. So, or in different types, we will come one by one. So in this lecture we will discuss about the phase 1 oxidation or phase 1 hydroxylation reaction of these different classes of compounds. Now when we talk about the drug molecules, when we talk about the chemical compounds that are generally we call it as a xenobiotics, they consist of diverse functional groups. And these diverse functional groups, as we have learned in the medicinal chemistry, that they generally have pharmacophores. Okay, so some of them, that the pharmacophores are the actual pharmacophoric identity that will give a pharmacological or any biological actions in the body. Now that diverse functional groups forces the body to adopt different mechanisms that it can render, the body can render the more hydrophobic drug to more hydrophilic drug, so that it can be generally easily eliminated from the body. Now since there are specificity of different kinds of functional groups, okay, so we can subdivide that oxidation process into many of these types, such as oxidation of aromatic carbon atom when the aromatic ring is present, when the carbon atoms are arranged in the form of aromaticity. When they show aromaticity, then aromatic type of hydroxylation occurs. So it is generally an oxidation reactions because it generally comprises the addition of the oxygen or the similar type of processes, or in case of oxidation of olefins. Now here the olefins are unsaturated compounds. Okay, so olefins generally contains the double bonds. Okay, so for that we have different mechanisms. Oxidation of benzylic carbon atoms. Now what is the benzylic carbon atoms? We will show it with specific example, as we have previously told that we generally describe each and every categories of these oxidations with specific examples of the drugs that we will discuss about. So you get a clear idea about what is the benzylic carbon atoms, how the mechanisms of oxidation takes place in kind of benzylic carbon atoms. So benzylic carbon atoms generally it is a carbon atom next to the benzene group. Okay, so there are different variants, we will discuss about that. In case of oxidation of carbon atom alpha to the carbonyls and imines, now it is a very important category of reactions. We will show with examples. In case of oxidation of aliphatic carbon atom, aliphatic means that the straight chain carbon atoms compound. Those drugs that consist of the straight chain carbon atoms, they undergo different kind of oxidations. Oxidations of alicyclic carbon atoms are also there. Also there are oxidations of carbon heteroatom system. In case of different classes of drugs, we will discuss that there are the bond difference between these. Not only the drug will act only on the carbon-carbon bonds, but they also act on the carbon heteroatom system. That heteroatom may be nitrogen, they may be oxygen, they may be sulfur. So according to that, we can divide the carbon-nitrogen system into N-D alkylations, oxidative deaminations, that means the separation of amine group. Okay, in N-D alkylation, so we will generally see that the amine group separates from the alkyl groups, okay, which is CH3 groups and multiples of that. And in N-oxide formations, we will also see N-hydroxylation reactions, Carbon sulfur system. In carbon sulfur system, we will study about S sulfur dealkylations, desulfurations, and L oxidations, S oxidations. Okay. In case of carbon oxygen system, we will discuss about O dealkylations. In case of oxidations of alcohol, carbonyl, and carboxylic acid, there are also different mechanisms. In case of apart from that, there are some miscellaneous oxidations such as oxidative dehydroxylations and oxidative dehalogenations. That means halogenations mean that the chlorine, bromine, iodine, all the halogens groups that are present. So for that kind of specific drugs, different mechanisms have been allotted. Okay. So, in case of oxidation of aromatic carbon atom, which is also known as aromatic hydroxylation reaction. Why hydroxylation reaction? Because of the addition of this hydroxyl group, OH group. So here what happens, since it is an example of aromatic carbon atom, so this is the structure of arene where it is monosubstituted with a substituent R. Okay. Now it undergo a highly reactive electrophile intermediate known as arene oxide. Now you can see that this is an epoxide linkage. Okay. So what happens that when this intermediate is formed, it is highly reactive. You cannot even find it in an analyte concentration. It is so much highly reactive. And when these arene oxide, in the absence of glutathione, which is also known as the natural antioxidant of our body. Why? Because of that, these have the glutathione, they have oxidant scavenging activity. Okay? They scavenge highly reactive oxygen species, ROS. Okay, so what happened in the absence of this GSH, generally these epoxide intermediate shows tissue toxicity. Okay, now this tissue toxicity can be carcinogenic or it can be cytotoxic. Okay, so this is a very highly reactive. Okay, so the retention time for these highly reactive arene oxide species or these epoxide species is so less that it undergoes simultaneously a fast reaction to produce this arenol. This is arenol. Okay. Now this arenol is a major product. Okay. So here the arenol, that is the para substituent of this hydroxyl group that is attached to this para position of this arene structure. So this is a major product, and also dihydrodiol. Okay, where the two hydroxyl group generally attach to this arene ring. Now what happened that ortho and meta product may be formed. It also formed in sometimes, but para product is the major. Here you can see this arenol product is the major. Okay. Now these undergo some the molecule gets retain its aromaticity and then it produces catechol. And since it is dihydrodiol, it is minor product, but the major product is the arenols. Okay. Now these dihydrodiols, they generally under the influence of GSH and 5-epoxide transferase, which is also an enzymes that is generally involves in such type of reactions that ultimately results in the glutathione conjugation. Now this glutathione conjugation, what happens that once it is conjugated with the glutathione molecules, it undergoes the phase 2 reaction. When we will study about phase 2 reactions, we will study in detail about the glutathione conjugation, what is actually the glutathione molecules, and how it attach to the drugs and carry out the later phase of phase 2 drug metabolism. We will study about that. So it is also a minor product that is formed in the presence of GSH. Now it should be present in our system in an appropriate concentration in our bodies to carry out this reaction. So there are certain drugs which undergoes such type of aromatic hydroxylation or oxidation of aromatic compounds. Okay, so these compounds are generally NSAIDs, that is non-steroidal anti-inflammatory drugs. Now what happens, this acetanilide, you can see this acetanilide is an NSAID. So what happened, that these undergo these para position hydroxyl group has substituted these. So generally acetanilide, which is inactive, it gets converted into paracetamol inside the body. So you can see that the paracetamol is produced inside the body. Okay, so in what mechanism? In these mechanisms only. Okay, in case of phenylbutazone, which is also an NSAID, what happens here? You can see also the para product. Here one thing is missing. Okay, here one thing is missing. That is, it should be OH. Okay, that is oxyphenbutazone. Okay, so here also the hydroxyl product is formed. In the next category, we have oxidation of olefins. Now, what is olefins? We have discussed that olefins is a non-saturated or unsaturated linear portion of the molecule. So, see here in case of carbamazepine, okay, we have two benzene rings, and in between them we have a linear portion. We have a linear portion with the double bond. Okay, so this portion represent the olefin portion of the molecule. Now this carbamazepine, they are used as an antipsychotic drugs generally. So in case of carbamazepine, we have this structure. Now what happens when it undergoes metabolism? It also produces an epoxide intermediate. Okay, so when it produces an epoxide intermediate, now what happened in previously we have also seen that in case of aromatic hydroxylations of aromatic carbons, there also the intermediate epoxide is generated. Here also the epoxide is generated, but in case of olephilic epoxides which is generated, which is the epoxide intermediate is much stable. That means it is reactive but not too much reactive to be cause so much harm. Okay, so what happened that generally these epoxide intermediate can be determined analytically. In an analytic experiments, you can determine this intermediate. So it undergoes further metabolism to produce the trans 10,11 dihydroxycarbamazepine. Okay, so what happens generally, the hydroxylation reactions is proceeded through an intermediate which is much more stable than that of the aromatic hydroxylation of aromatic carbons. Okay, so most stable intermediate than aromatic hydroxylation can be detected analytically. Now there are exceptions, like all other experiments there are exceptions also. In case of olefin epoxide of aflatoxin B1. Now what is aflatoxin B1? It is a toxin produced by Aspergillus flavus. This Aspergillus flavus, it is a type of saprophytic fungus. Now what happens is aflatoxin B1, they are potent carcinogen. Okay, they causes carcinogenesis within a very minute amount, and this generally causes hepatic cancer. So in case of olefinic epoxide of aflatoxin, these cannot be determined analytically. Okay, so these are highly reactive. So this causes hepatic cancer. So in case of oxidation of benzylic carbon atoms, now what is benzylic carbon atom? Generally the benzylic carbon atom is the carbon atom just next to the aromatic portion. That is this carbon atom next to the aromatic system, it is the benzylic carbon atom. Okay. Now in case of tolubutamide, okay, in case of tolubutamide, or it is sometimes known as tolubutamide, it is generally sulfonyl urea group of anti-diabetic drugs. Okay, so these generally sulfonyl urea group of anti-diabetic drugs, they generally undergo metabolism by this hydroxylation reaction, that is OH group, that is attached to the benzylic carbon of the system. So it undergoes the under the presence of alcohol dehydrogenase enzyme, it is converted to subsequently aldehyde, and subsequently the corresponding carboxylic acid is formed Okay, so generally this is another mechanism when we got the benzylic carbon atom next to the aromatic portion of the molecules. We in our body generally this type of metabolic reactions go on. Let us see what happens in the oxidation of allylic carbon. So what is let us consider what is allylic carbon. Okay, so allylic carbon is the carbon atom next to the double bond. Okay, so here you see in the structure of hexobarbital, which is also a type of barbiturate drugs. So here you can see that the carbon atom that is next to the double bond. Okay, so this 3 dash, this is the alicyclic carbon atom. Okay, now this alicyclic carbon atom, what happened that during the hydroxylation reaction or the oxidation of such kind of alicyclic carbon atom, the hydroxyl group just attached to this 3 dash carbon atom. Okay, so this type of that results in the formation of 3-hydroxy hexobarbital as a metabolite. So this kind of, so you can see that ultimately the addition of the hydroxyl group to this portion, this reduces the lipophilicity, this increases the hydrophilicity and reduces the lipophilicity, ultimately rendering the drug more lipophilic. Okay, rendering the drug less lipophilic and more hydrophilic. Okay, so in case of oxidation of carbon atom alpha to the carbonyl and imino, that is CN group, this special kind of oxidation is we can see in the kind of diazepam, which is also a type of generally it is sedative. Okay, so sedative and hypnotics we can study diazepam. So here we see what happens is that the hydroxyl group is attached to the carbonyl, the carbon number that is alpha to the carbonyl group. Okay, so here you can see that the CO group is here in this structure. The CO group is here, and you can see that the carbon atom that is alpha to the CO group is this carbon atom, and also the carbon atom that is alpha to the imino group, that is here it is C, here it is N. So in this case also it is 3 dash. Okay, so here what happened that this carbon number generally gets the addition of a hydroxyl group. So it leads to the formation of 3-hydroxy diazepam, that is also known as N-methyloxapam. Okay, so this is the case of diazepam. Okay. So in case of oxidation of aliphatic carbon atom, we generally consider as a these types of oxidation reaction is generally seen in case of anti-epileptic drug, that is valproic acid. We generally used in case of status epilepticus. Okay, so what happened? The much more linear structure is present in the molecule. Okay, so what happened that this contains the terminal CH3 group. Both this branch contains the terminal CH3 group. Now here the 2 types of oxidation is possible. Okay, one is known as omega oxidation, where the hydroxyl group is attached to the terminal CH3 group, rendering it to the formation of 5-hydroxy valproic acid. And another is the omega-1 oxidation, in which the carbon number that is penultimate to the terminal CH3 group. So what is the carbon atom that is penultimate to the CH3 group? So here you can see, you can observe here that in the CH3, this is the terminal methyl group. So the carbon number that is penultimate to this is this carbon. Okay, so the hydroxyl group will ultimately attach to this carbon atom, leading it to the formation of omega-1 oxidation. So there are 2 possible oxidation product of this valproic acid. So in the next lecture we will see more another 2-3 more example or 2-3 more categories of these oxidation reaction, and also in the next lecture we will possibly we will possibly discuss the other form of phase 1 reaction, and we will ultimately go to the phase 2 reactions. There is a message for you guys. One of our friends, Rahul Sarkar, he has started a new channel about the BDS support. which we can provide you lots of informations about the BDS subject or dental subjects, and those who are willing to study his subject can follow this channel. And generally those who are pursuing BDS in the final year or the juniors, they all can follow his subjects. If you can follow, it's very informative. It will, if you can follow that channel, then it will be very informative to you. So please subscribe his channel of BDS support. The description of the link is given in the description box. So please follow and like it.